Today I have the pleasure of presenting Dr. Mark Henderson, who, as Eric just said, is our last seminar speaker for this year, or for this spring. Mark is coming here today from Humboldt State University, where he's the leader of the California Cooperative Fish and Wildlife Research Unit. His burgeoning lab includes research on everything between deep sea corals to oyster aquaculture in Humboldt Bay, though today he presents juvenile Chinook salmon mortality in different habitats during migration from freshwater to the ocean. Just give him a round of applause now.
Thank you, Laura. So I sat on Laura's committee, so I've known Laura for a while when she was at Humboldt State University. I expected a much more embarrassing introduction based on what I know of Laura from her time with me. So thank you for a very kind introduction. I appreciate you guys all sitting so close. If this was a lecture, I would probably go to the closest person to me and try to give a lecture like this to try to embarrass people a little bit more, but I'm going to be nice and I'm going to stand a little bit farther back. So what I'm going to talk to you guys a little bit about today is some of the research that I've done about salmon. I realized that since I'm so used to giving this lecture to people on the West Coast who are extremely familiar with salmon and who have seen the cycle of anadromous salmon so many times that I didn't actually include any cycle of the life cycle of the anadromous salmon, so I'll probably have to talk you through that a little bit, but I'm assuming that most people have probably seen the life cycle of anadromous salmon. But anyway, we'll get to that when we get to it. Just to give you a little bit of my background, I did my PhD at the Virginia Institute of Marine Sciences before I moved out to California. Before I moved out to California to start a postdoc doing life cycle modeling of salmon in California, I didn't actually know that there were any salmon in California. I was surprised to find out that there were salmon in California. I didn't realize that they went all that far south. And the reason for that is because most of the salmon that are in California are either endangered, threatened, or species of concern. And the reason for that is because well, before I get to that, I just want to acknowledge all of my collaborators that helped me out with the different projects that I'm going to be talking about today. There are people from UC Santa Cruz, one of my grad students, Chris Loomis, who's from Humboldt State University, Dave Huff from NOAA, Licia Iglesias from Santa Cruz, and Joe Smith. And I couldn't have done any of this work without them. They contributed just as much, if not more, than what I did to many of these projects. So I'm going to be talking about two main projects today. One is a paper that just recently came out in the Canadian Journal of Fisheries and Aquatic Sciences, estimating salmon outmigration mortality through the Sacramento River. And then the second project is once they get out of the Sacramento River, estimating predation risk once they get into the delta and then before they get out into the ocean. So, like I was just about to say, so once they, so this is the range of of Chinook salmon. So all the way from Kamchatka, Russia, all the way through Alaska, down through the Pacific Northwest, and down through California. So the areas in green are their current spawning distribution. The areas in red represents where their historic spawning distribution is. So you can see down here in the south, especially down in the south, they have relatively limited, or their historic spawning distribution is more limited. And the reason for that, and this is where we are down in California, they have some more limited spawning distribution to where it used to be. And the reason for that is mostly because in California, dams have been put up. So a lot of these areas, this area in red or brown as it looks up here, is what's been blocked off. This is what's been blocked off from their previous spawning distribution. And the reason for that is that there have been a lot of dams up here because, as you may know, water is big business in California. There's been dams put up so that they can save water and so that they can ship water from various areas in California to other areas in California for agriculture. There's If you drive through the Central Valley, which is this area, so this is the Sacramento River up here, you'll see signs everywhere that say fish over farmers. So they'll, a lot of times they'll limit dam releases, and the farmers want that water. They'll limit the dam releases so that they'll save water, or they'll have different dam release schedules so that there's enough water for the salmon, but the farmers want to save that water so that they have enough water for their farms. So the farmers say that you're killing our fields because we need that water for our farms, but then the salmon need that water. And then in the delta, there's another endangered species called the delta smelt that they need that water as well. So there's all these water rights. And then once you get down into the delta, and we'll talk about this a little bit more, there are other export facilities down the delta where they take all this water that comes down from the Sacramento River, and then they ship it down even farther down to Southern California, so that there's water down there for LA and all the areas down in Southern California, so that all the farms down there have enough water for them to grow almonds and other areas like that, or other crops like that. 90% of the almonds that we eat in the United States are produced in California, and it's an extremely water hungry crop. And they don't actually, when we go through droughts, like we went through some of the worst droughts in history, well, some of the worst droughts in a long time in 2014, 2015, they were losing a lot of the trees, a lot of the almond trees down there, and they were getting very angry about some of the water issues that were going on. So basically water is a big deal in California, is the point that I'm trying to make. So they've lost a lot of this historic spawning habitat because of these dams. So because of that, these salmon species are now listed as species of concern or threatened. So for this talk, we're going to be talking about fish that are in the river portion, the first part, and then fish that are in the delta. And then once they outmigrate, they also have to go through the bays, but we're not going to be talking about that. And then obviously they're going to be in the ocean as juveniles. So they're anadromous, and then they come back as adults to spawn. So there you go, there's their life cycle. So in a paper by Cyril Michel, one of my co-authors in 2015, he found that the survival per 10 kilometers in the river, the delta, and the bays was, if you look at survival in the river, it was lowest in the river until this one year, and this was 2011, and it went way up. So you can see that the variability in the delta and the bays was fairly consistent, but in the river there was this huge variability. And the reason for this variability is that in 2011, that was a really high water year. And so there was a lot of flow in that year. So what the conclusion from that is that high water year was really good for salmon survival. So what his conclusion is that if we want to help these salmon survive, the area that we should probably focus on is the river. So figure out what's going on in the river, and then we can hope to maybe mitigate their survival in the river, figure out what factors are affecting their survival, and that's the area that we can focus on to maybe help their survival. So this is one of the areas that we try to focus on. So what are some of the potential areas of concern? What are some of the potential factors that could be affecting their survival as they're migrating out through the river? So we have some of the water management issues that we were talking about, the dams. We have loss of habitat complexity. So in the lower half of the river, because of trying to make the river more navigable, they channelized a lot of the river. So they put a lot of riprap and things like that throughout the river. There's been a reduced flow because of dams and because of reduced precipitation due to climate change and things like that. Predation from a lot of introduced species like striped bass that have come into the river. increasing temperatures due to climate change, non-native species from both fish and plants, such as this water hyacinth. Entrainments, so diversions that are from the agriculture facilities that have been taking out waters. It's basically a bunch of straws that go into the river that are just sucking out water. And they have water rights that go back to the 1800s. And once they have those water rights, you can never take them away, basically. Poor water quality due to temperature and agriculture runoff. Loss of floodplain habitat, so once some other studies have shown that some of the growth of these fish is higher when they're in the floodplain habitats. So naturally, when the flows are higher, they move out onto these floodplain habitats where it's shallow and warm and there's really good food, so they grow really big. But they lose a lot of this floodplain habitat when it's channelized. And then riprap, so kind of loss of habitat complexity. That's all tied together. So the objective for this study was to determine which of the environmental, physical, and biological factors are having the greatest influence on their outmigration survival. And I'm just going to focus on two of some of the major findings that we had in this study, which are flow, and the major question is, more flow always better, and release strategy. So we're working with hatchery fish here. So if we're releasing, what release strategy should we use when we're working with hatchery fish? Should we release a lot of fish all at once, or should we release them in small batches, multiple small batches, so that if we release them throughout multiple small batches throughout time, are we going to have kind of a portfolio effect where if we release multiple small batches, they're going to have an increased chance? So the way that we did this, the tools that we used to do this, is we used acoustic telemetry. So we used a tag called JSATs, or Juvenile Salmon Acoustic Telemetry System. So this is a comparison between a JSAT tag and a PIT tag. So the PIT tag is what they typically use for juvenile salmon tags. So you can see it's not much bigger, it weighs a little bit more. But we can put those into salmon that are about 60 millimeters into their peritoneal cavity. Sorry, I lied. That is the size that we can put in pit tags that we're pit tagging on rivers. We're putting these into fish that are about 120 millimeters. Sorry, so twice as big as that. And then once they go down and they pass by these acoustic receivers that we have in the water, we can hear when they go by the acoustic receivers. So then we can plug these data. So here's the length range of the fish. And here's where we have the acoustic receiver gates throughout the entire Sacramento River. So we had 19 gates down through the river. So we were working with 5 years of data from 2007 to 2011. We tagged 1,706 hatchery smolts. And so we listened to them as we go. And then once we had those data, we could plug them into a Cormac Jolly-Sieber model. And then, so for those of you who aren't familiar with this type of market capture model, it's basically a multinomial model, so that fish who's released, when they're released, they have two possibilities. After they're marked and released, they could either survive or they could die. A fish that survives could then either be captured or not captured. A fish that's captured would have an encounter history. This is the first encounter when they're marked and released. So all fish that are marked would have a 1 in the first occasion when they're marked and released. And then this is the second encounter. So only the ones that are captured would have a 1. The ones that are not captured or die would both have 0s. And then as this happens throughout multiple occasions, remember we have 19 occasions, they're going to have multiple 1s and 0s as you keep on going out. And then when you put that into a multinomial framework, then you can estimate each of the parameters for survival and capture throughout that entire history. And that gives you survival and capture probabilities throughout the entire time frame. So then once we have those survival and capture probabilities, we can also put this into a covariate framework. So we can put our covariates that we're interested in, so we can get a kind of mechanistic understanding of what covariates are affecting these survivals. So, which is really what we want to go. We want to get a more mechanistic understanding of what's affecting the survival of these fish as they're migrating down the stream. So we had 3 different types of covariates that we were putting into these models. We had individual covariates, so the transit speed, the speed that fish were moving through each of these reaches, whether they were released with a bunch of other fish or whether they were released in smaller batches. And then some reach-specific covariates, so the habitat-related covariates. So whether there were lots of diversions, or diversions are the straws that were into the river, how much water was being sucked out by these agricultural diversions. The sinuosity, so that's a measure of habitat complexity. So is it a more natural channel that's kind of meandering along, or is it kind of a straight channelized cut through the river? And release mortality, so was that where the fish was released or not released, expecting that when fish are tagged and released, they might have a higher tagging-related mortality in the reach right after they're released. And then time-varying covariates, so flow and water temperature, which are going to change through time. So we incorporated all of these covariates into the model. Once again, these are the 2 covariates that I'm going to be focusing on for the purposes of this talk. So in order to get to those time-varying covariates, we coupled this model to a physical model, which is the river assessment for forecasting temperature, or the RAFT model that was developed by a colleague of mine named Andrew Pike. And it estimates water temperature, flow, and velocity every 2 kilometers down the Sacramento River. So this is a time series for one particular location along the river called Ball's Ferry. This is the measured at a gauge, and then this is the model predicted temperature at a gauge, just giving you an idea of how that works for one particular location. And then if we put this all together, so this is the distance from Keswick Dam. So Keswick Dam is up here. This is 100 miles down from Keswick Dam for every date from May 22nd all the way through October. You can have a prediction for every river kilometer for every day at every location. So now we have a complete time series for every location at every spot. So we can go in there when we know where the fish is and when the fish was there. We can go in and we can extract that exact piece of time, and we can extract that temperature, and we can plug that into our model to say, okay, when the fish was here, this was the temperature that they were experiencing, and plug that into our model. So then we compared 2 models to see which one was fitting better. We compared a reach by year model, which means for each reach in each year, we estimated a different survival, and that had 110 parameters in the model. And then we had this kind of mechanistic model, or a process model, where we had those mechanistic variables. So that only had 15 parameters. And then if you look at the delta AIC, we use model selection to compare them. Delta AIC, 56, 0. So obviously this process model is fitting the data is a much more parsimonious model. So it's a simpler model. It's representing the data on much fewer parameters and still fitting the data reasonably well. So I'm just going to walk you through this goodness of fit just to give you an idea. So, and then I'll show you, kind of expand it to all of them. So this is comparing those 2 different models, the reach by year model to the process model for all the different years, for all 5 years. So when the process for these positive deviances, this means the process model overestimated mortality. For the negative biases, that means the process model underestimated mortality. When the 95% confidence interval spans 0, that means there's no significant difference between there's no significant difference between the 2 different models. And once again, this is for all 5 years. So we did it for every one of the 19 different locations. And kind of the take-home message from this is at the lower parts of the river, so up here is kind of closer to Keswick Dam, down here is closer to the Delta area. So at the lower areas, there's less differences between the process model and the reach by year model. The bigger differences are up here, especially in the earlier years. So what that means is that it's in the earlier years where we seem to be missing some covariates in our process model. So what are those potential covariates that we might be missing? So our model, we don't have any measurements of large woody debris. We don't have any measurements of substrate type. We don't have any measurements of predation. We didn't have any independent samples of how many predators were out there at the time that we were doing these samples and fitting these models. And we didn't have any measurement of turbidity in our model. Unfortunately, that wasn't included in that physical model that we were trying to fit. So we think that if we could get some independent measurements of these into our model, that would definitely improve our model and improve our fit and reduce our uncertainty. So we're trying to encourage the California Department of Fish and Wildlife to start getting some independent samples of these and get some more independent surveys so that we can incorporate those sorts of things into this model. So kind of reviewing what we had, is more flow, so going back to our objectives and our results, is more flow better? So what we found is that there was an interaction between flow at one particular location. So this is annual flow at one particular location. So, and then the flow within a reach. So as annual flow increases, survival increases. But then we also found that it was whether or not flow was above or below average within a reach. So I need to explain that one a little bit more because it's a little bit confusing. So if flow is above average within a reach, you can kind of see that that line is a little bit more horizontal. So what that means is if flow is above average within a reach, it doesn't really matter as much what the annual flow is. So if there's a large precipitation event, so if there's a large rain event and it comes after a storm or something like that, it doesn't really matter what the annual flow was for that year. If it was a lot of flow or a little flow, it doesn't really matter. Survival is going to be good no matter what. But if it's a low flow year, so if it's a drought year, then it really does matter if it's below average flow, or the annual flow really does matter if it's below average flow. Basically, what it's saying is precipitation events matter, or precipitation events matter when it's below average flow for the year. It's kind of what happens. In drought years, precipitation events matter, and they don't matter as much when it's above average flow. It's kind of the simple way of saying that. And then release strategy, single large release or multiple small releases. We found that there was an interaction between distance from Coleman and whether or not there was a batch release. So when there was no batch release, survival was still pretty high. But when there was a batch release, we found that survival was even higher, meaning that there was probably some predator swamping going on so that they were not getting eaten or dying quite as much. But that only lasted for about 200 kilometers downstream from where they were released. So conclusions, we could reasonably reproduce the spatial and temporal smolt survival, but that could be improved with additional covariates. Flow was important to survival, but more flow only mattered in low flow years. And evidence of predator swamping with large releases, but this effect was negligible after about 200 river kilometers. Okay, so now moving on to the second part of the talk, which is estimating predation mortality in the Sacramento-San Joaquin Delta. Remember, we just moved out of the river portion, so now we're into the delta portion, and this is kind of a close-up, or this is a close-up of the delta area. So as you can see, we're moving out of this kind of large, very channelized area, and now it kind of gets much more, there's much more distributaries. It kind of goes into this very diverse network of channels throughout here. And this is an extremely modified from what it was originally and naturally. Where am I on time?
You've got 20 minutes or so.
All right, cool. So the San Francisco estuary has been called the most invaded estuary in the world. And that is both fish, so striped bass, which has also striped bass. So Peter Moyle, who's a big fisheries biologist in California, has now called striped bass an honorary native species because it's been there for over 100 years. And nobody would ever want to get rid of striped bass. Striped bass, largemouth bass, catfish, those have all been introduced. These were mostly introduced in the late 1900s by the California Department of Fish and Wildlife for recreational angling purposes. Corbicula, the Asian clam, mostly came over in ballast water. Then there's introduced copepods that have come in. These have completely changed the food web within the bay. So as a result of that, there has been something called the pelagic organism decline. So things have completely changed within the different fish species have changed throughout there. There's because of the corbicula, they basically suck all the phytoplankton out of the water. Complete opposite problem that Chesapeake Bay has. Chesapeake Bay is eutrophied. The San Francisco Bay is basically completely clear because corbicula is sucking all the phytoplankton out of there. So there's not enough food for the zooplankton to eat, so there's not enough food for the fish to eat. And so the fish, there's not enough food for the small fish to eat, so there's not enough food for the big fish to eat. And then we have invasive plants like this. This is submerged aquatic vegetation. It's a Brazilian waterweed. And so I'm going to be talking much more about these fish, and I'm going to be talking much more about this waterweed. It's going to come back throughout the rest of this presentation. So just to give you an idea of the comparison between non-natives and natives, this is from a paper by Thayer and Healy in 2003, where they went out and they did a survey. This is the catch per unit effort of non-natives versus natives within the bay. There's a lot of non-natives. So, so why do we care about survival and estimating predation risk as they're migrating through the delta? So there's been a bunch of acoustic telemetry studies looking at salmon. Once again, people really care about salmon in California. It's $1 billion business for salmon fishermen in California. They care a lot about it. So even though they're endangered or threatened, the fall run of salmon is still a commercial fishery. There is still a commercial fishery for fall run salmon, and they're hoping to bring back some of the other runs. There are 4 runs of salmon, fall run, late fall run, winter run, and spring run. And they are hoping to bring back some of the other runs so that eventually they do support other fisheries. They actually, historically, had some of the largest runs on the West Coast, as large as Columbia River. So, or at least that's what some of the evidence shows. And acoustic telemetry shows that survival through the delta is extremely poor, especially if you take some of the routes, so the interior delta. So survival through the main stem Sacramento, which is what these routes are showing, actually isn't so bad. They can do fairly well if they take those routes. But if they end up going through this interior delta, it's basically a death sentence for them. Some of the evidence has shown that their best chance of surviving if they go into the interior delta is if they end up down here, which is where the export facilities are, which I mentioned is the water that gets shipped down to Southern California. Because what happens is if they get into the export facilities, they get trucked out to the delta. So that's the best way for them to survive, is if they get trucked out. So yeah, so basically they get in there, they get eaten, is what happens if they end up in there, because it's a maze, they can't find their way out. So, so what this has shown is that survival is much less than any of the other West Coast estuaries. And predation is primarily what's suspected to be the primary culprit for this really low survival rates. One modeling study from Steve Lindley and Mike Moore has shown that for winter-run salmon, striped bass could eat enough salmon. The striped bass population, this is for 3 different levels. This is without stocking. This was back in 2003 when they were still stocking striped bass. They've since stopped. But this was without stocking with moderate levels of stocking, and this was at their current levels of stocking. That this was how long it would take to cause extinction. So without any stocking, they said that it would take 50 years for straight bass to wipe out the winter run salmon in San Francisco Bay. So what are some of the key questions that we have for salmoniid predation in San Francisco estuary? One is how many predators are there within San Francisco Bay? And you might be wondering what are those predators? Predators are primarily the striped bass, largemouth bass, and smallmouth bass, white catfish, and channel catfish, so all introduced species. What environmental factors are related to these predator distributions? And then, are these same factors associated with salmon predation? So the study that we designed for this, this is actually the second iteration of this study, and I'll talk a little bit about some of the lessons that we learned from the first iteration. Started in spring of 2017. We selected 21 sites, including 3 repeat sites. And we sampled over 6 consecutive weeks during the main salmon, juvenile salmon out-migration time period as they were going through the delta. And this was in the southern portion of the delta. We couldn't sample the entire delta, it just wasn't logistically possible at the time. We used 2 different tools in order to do that. And the way that we randomly sampled these sites was something called GRITS, or Generalized Random Tessellation Stratified. So this is a spatially balanced random sampling tool. And the way it works is we split our area up into 8 different areas that we felt had similar hydrographic conditions, and then we spatially sampled each of those 8 different areas and equally sampled our sites within each of those areas. And so this GRITS kind of spatially balances the sampling within each of those 8 different areas, independent of the area of those 8 different areas. So the tools that we used to sample, we used DITSON, or dual frequency identification sonar. We used 2 DITSONs mounted on opposite sides of a survey vessel that we ran transects down the channels on either side. And I'll show you an image of what this looks like. But basically, it's a hydroacoustic method that gives you video-like quality of the fish that you're looking at. So it gives you a more accurate survey method than kind of a traditional hydroacoustic survey, which is what we originally used when we did our kind of pilot survey in 2014, 2015. And then the second tool that we use is this tethering project, or this tethering tool that that we developed called the Predation Event Recorders. So what's on these Predation Event Recorders is a GPS transponder and a tether that we tethered a juvenile salmon smolt to. And this salmon smolt, this tether was attached to a magnetic, or to a magnet basically, that when it was pulled out, a timer went off so that we could figure out exactly when that salmon was predated. And then we could figure out exactly when that salmon was predated, and then with the GPS transponder, we could figure out exactly where that salmon was predated. So, lessons learned from the traditional acoustics. We did this project, we started this project, it was a large project in 2014 and 2015 with traditional hydroacoustics. We thought this was going to be a really good idea because traditional acoustics could sample a much larger area, much faster than Ditson. But the problem is this is what the riverbed looked like, and these are what the fish look like. So we could get some idea of a count of what the fish were, and we could get some correlation of the fish to the predation events that were happening, but the surveys of the excuse me, do you have my water?
It's on the ground.
Yeah. But the surveys were not happening at the exact same time that the PERS experiments were happening. I'm going to be referring to the predation event recorders as PERS because it's easier. So they weren't exactly happening at the exact same time. And we couldn't get a completely accurate measurement of the size of the fish because we were relying on target strength, which is the amount of sound that was bouncing off of the fish in order to, so a target strength to length relationship, which there's a lot of uncertainty involved in those relationships. So we couldn't get a completely accurate measurement of the size. So basically what we found in 2014 and 2015 is that there was no relationship between predator density and predation risk. So, but we didn't believe that. We didn't think that that was really accurate. So we went out and we used Ditson. So this is a video of Ditson. And you can see up here, these are fish. And these are probably striped bass going by. And you can actually measure these fish as they're going by. You can stop each frame and get a pretty accurate measurement. And while we were doing this, we also did some electrofishing experiments. We stopped, we took some fish, we tethered fish, and we tethered large predator-sized fish. We insodified them with the Ditson, and we took some length measurements of those tethered fish, and they were actually really accurate, the length measurements. So we can get both length measurements and we can get width measurements, because you can, with these hydroacoustics, you can look at them both sideways or length on, and you can look at them on top of them because sound goes around things. So we had really good measurements, and we could do these surveys concurrently with what was happening with the PERS. So we were running these Ditson transects at the exact same time that the PERS were out there. So we had high target resolution. We could get our morphometrics. We could observe the behavior of the fish. It was visually intuitive. We were also, because of the way it was, we could see the bottom. So we could see some sab. We could see if there was any, if there were tires or things along the bottom. We could see that with the Ditson much better than we could see with the traditional acoustics. So this is just kind of some preliminary results of what we saw with the Ditson. This is a comparison from 2 different dates. By the way, this is all work from my grad student, Chris Loomis, who is depending in a month, a little less than a month. And so here is from April 27th and then May 8th. And so you can see 2 different densities. This is 52 fish and 248 fish. You can kind of see the difference in density between those. So just kind of showing you what it looks like with two different densities of fish. The two different colors just represent two different transects from when they were going through the reach. So now that we have these data, we also collected a whole bunch of different habitat variables as we were going through there. Temperature, depth, various things. So we fit a whole bunch of models, generalized linear models, to figure out which habitat variables were actually the best predictors of where these predators were, because that was one of the other questions that we were trying to figure out. So from that, we did our model selection, and we found that the coefficient of variation of depth, a measure of habitat complexity, sinuosity, another measure of habitat complexity, and SAV patches, remember, that's the invasive species that I showed on that plot on invasive species with that person's hand covered in weeds. So the number of SAV patches, so not the amount of SAV, but the number of SAV patches were all predictors of predator probability of predator, or predator density. So here are the response plots showing the relationship between those, between the predicted predator density and those various variables. So sinuosity as you go down, which is actually the opposite of what we expected. All we can think about is that maybe those were in, so I'm going to explain the one that we don't understand first. So maybe those were in channelized habitats where there were some riprap, things like that, fishermen tend to stand on some riprap habitats and fish off of those. But coefficient of variation of depth, so in the more complex habitats, they tend to like, predators tend to like being in more complex habitats. And then SAV patch density, these tend to be mostly ambush predators. So they're hanging out, especially largemouth bass are more associated with more complex, so they're hanging out on the edges of these patches. And we also, I should say that also in this model selection was the abundance of SAV, and that didn't come out in the model selection. So it was these patches. They wanted more patchy SAV habitats where they could hang out on the edges and then come out and eat the smolts. So from that, we now can predict. So we didn't sample this whole area, remember, we randomly sampled 21 sites within that. But now that we have this model, we can now predict predator density throughout the entire South Delta. So this gets to that second part of our question. What is the predicted predator density throughout this entire South Delta? So now, I'm just going to highlight a couple of areas. So this is Clifton Court. This is one of the export sites. And this is one of the areas that they try to divert fish away from because they think that there's going to be a high density of predators. And there is a relatively high predicted density of predators near Clifton Court. But the highest density of predators appears to be at the head of the Old River, which is where they tend to divert fish away from Clifton Court to go to the head of Old River and stay in the main stem, San Joaquin. So one of our conclusions from this is maybe that might not be the best management strategy. So we'll see what happens when the managers see those predictions. And remember this head of Old River, because that'll come back in the second part where we'll be talking about are the same factors associated with salmon predation. So now when we try to estimate predation risk, so our objective is to use these predator abundance that we just estimated with Chris's thesis to elucidate the variables that govern predation and estimate the spatially explicit estimates of predation risk. So now we're using these tethers that I talked about. So we're using Cox proportional hazard models. And Cox proportional hazard models, if you're not familiar with them, these were developed by the health sciences basically they're disease models to figure out time to death, basically. So if you have different people with diseases and they're dying at different times, and some people are getting censored, you want to figure out what are the factors that are affecting how long it takes different individuals to die. So we have a similar sort of situation here where we have some fish that are getting predated, some fish that aren't getting predated, some fish that are getting predated sooner than others, and we have this time factor that's getting factored in here that we need to incorporate into our models. So, but we also have this mixed effect, this random effect that's in our model because we have sight in there, and that's a random effect. So we include that within our model, and we can handle right-censored data because there were some PERs that made it all the way through each of our 1 kilometer reaches without ever getting predated upon. So we need to be able to handle that data, and these models are able to handle this. So these are the variables that we included in there. We have temperature, turbidity, depth, the coefficient of variation of depth, submerged aquatic vegetation, distance from shore, predator density. This is what we just talked about with Ditson. Time of day, typically pretty much every fisherman knows the closer you get to sunset, the more likely fish are to get eaten. They like to eat at that magic hour right after sunset. And water velocity, which we use as the speed of the PER moving through the reach. So before, what we were talking about was the density of the predators within a reach. Now what we actually want to do is relate this to the actual space where the PER was at any given location. So instead of using the density of the PER throughout the entire reach, what we're actually going to use is a nearest neighbor approach. So what we do is we take the location of the PER at any one particular point, and we do this at every point where a PER is, and we find the 10 nearest neighbors, and we calculate the distance to the 10 nearest neighbors of that PER, and then we calculate the mean distance to the 10 nearest neighbors. So basically that gives us a mean distance to predators, and that gives us an idea of how close predators are to a PER at any one particular location. So that gives us an estimate of predation or risk of a predator being close to that PER at any one time. So here are the results from that. We went through the same model selection process and found that there were 4 prime variables that were predicting predation risk. The coefficient of variation of depth, once again, habitat complexity, water temperature. So the way to read these variables is if you're above 1, it means you have a higher risk of predation. If you're below 1, it means you have a lower risk of predation. And then distance to nearest predator, and then time of day. So as you get close, so after sunset, you have a much higher risk of predation. As you're closer to a predator, you have a much higher risk, or your mean distance. The closer you are to a predator, the higher risk of predation is. The higher the temperature you are, the higher your risk of predation. So you can kind of think of that as the higher the metabolic demand of the predators that they are, the more likely they are to try to eat you. And the coefficient of variation of depth. So the more complex the habitat is, the more likely you are to get eaten. That predators like complex habitats, so since they're mostly ambush predators, you're more likely to get eaten in a more complex habitat. So this is just a animation showing through the season for each of the 1 kilometer reaches the risk of getting predated in each one of those 1 kilometer reaches through the season. And as you can see in the early part of the season, when the temperatures are relatively low, risks are relatively low. It's pretty much as you get later in the season when the risks start to go much higher. And then right around now, they go pretty high. And then it'll start over. So, so it's pretty much what it's driven by mostly is temperature and distance nearest predator, and then time of day. So I don't know what time of day it was. I'm assuming he picked, it was Cyril who made this image, and I'm not sure what time of day he used. So just as an exercise, he went through, this graph is also made by my colleague Cyril. He went through and he picked the sites where the percent of daily hazard ratios were above 1 for the spring of 2017. And he wanted to see if any sites stood out. And he found that there was this one site right here where 100% of the time, the percent of the daily hazard ratio was above 1 for the entire spring of 2017. And it turns out that this site was, once again, the head of Old River, which was the same one that I talked about before, where we were diverting fish away from Clifton Court forebay down to the head of Old River so that they could avoid predation or avoid getting entrained into the head of Old River or into Clifton Court forebay. So once again, recommendation to managers maybe that might not be the best strategy to try to avoid fish mortality by going down Clifton Court forebay. So the conclusions from this part of the story is that invasive submerged aquatic vegetation was associated with higher densities of predators. So one of the recommendations that we're making to our managers is that maybe we really need to kind of ramp up our removal of this invasive submerged aquatic vegetation. They've already started some of this, but that really needs to be kind of, prior to this, one of the primary salmon predator control strategies was eradication. So trying to remove these predators. So removing bag limits and removing the predators. And we think that maybe targeting the habitat rather than targeting the predators may be a better strategy. And our preliminary results suggest that our predation risk model is accurate, at least based on that head of Old River. That's a pretty well-known predator place where people go to target predators, fishing location. And if we can really perfect this model, well, not perfect, but keep on working on this model and calibrate this model, we think it's going to help managers to predict where salmon risk is increased and help manage the releases and reduce at least hatchery releases as they're moving through there to release their mortality risk as they're migrating through. So future work, we recently submitted a big proposal. So this was the area that I was just talking about, the South Delta. We submitted a big proposal. We want to do a whole bunch of work in the interior Delta, North Delta, and the Sacramento main stem to compare, to do predator surveys and combine some molecular techniques and some standard survey techniques to look at predator distribution throughout the entire Delta. I'm happy to talk to people about it. I think it's going to be a really cool project. And then I just want to acknowledge all our different funding sources, all the people who helped us out with this, including especially the people who helped out with the Ditson. These are some pretty fancy fish. And then with that, I'll take any questions. I lost my questions. Yeah.
So I think the mortality or the survival probabilities you were showing in this first piece of your talk were daily survival probabilities. These were on 0.95, 0.97. Is that?
Those were per 10 kilometers.
10 kilometers. And then how long does it take to a fish to transit the 10 kilometers?
They can do it super quickly. It's a matter of hours.
Okay.
Mm-hmm.
Yeah, just wondered if, I mean, if you pinpointed or, you've described and then your colleagues have described these areas of high mortality. It's such an altered system. They're trucking fish anyway. I mean, why not just release them down much farther along what's the downside of trying to avoid running this gauntlet of predation and
because they're trying to improve the natural population coming back to spawn, so they want them to come back to the hatchery to spawn. So if they've tried trucking before, and the number of fish that come back to so they need the smell of their natural habitat in order to find their way back home.
So too much straying if you leave them.
Exactly.
If you put them down too far.
Yep.
Yeah. So there's probably some sweet spot there. Yep. That's probably variable in time too, yep. Depending on what the strength of the river flow and those dynamics
mm-hmm.
are.
Yep. Exactly.
Yeah. Thanks.
Yep.
Another question? This Stinson thing.
Yeah.
How clear does the water have to be, or how turbid before that goes bad?
So it's that's the great thing about the acoustics. I mean, so I think there is a turbidity where it won't work, but I think it's beyond anything that you're going to see in. It'll work in almost any turbidity.
I was wondering whether it would work in Mississippi Sound.
You could try it. I mean, the Sacramento is pretty turbid, and it works in there.
I was just wondering, so you found that the area where they're diverting fish to is where they have the highest predation risk. Do you think that those are interrelated, like the fish are going there?
I asked Chris the same question. Is it the chicken or the egg? Are they going there because the fish are getting diverted there, or are they? Yeah, I don't know the answer to that. And yeah, nobody knows the answer to that. It's a good question. Did you have a question?
So I'm curious with removing the submerged aquatic vegetation, kind of like if that is feasible in that region to continually harvest or remove the submerged aquatic vegetation, do you presume it would cause the predators to move to a different area, or more so just be more visible to those prey to where they aren't able to be as hidden and ambush as well, or would they move to a different area, do you think?
It's a good question. So so one of the other things we found is that they tend to associate with structure or another. So if they didn't associate with Sav, submerged aquatic vegetation, they'd probably associate with tules, which is a more natural vegetation. So they would probably move to another area that had tules, or another man-made structure like some diversions or something like that. So yes, but if we could move them more to the area that we wanted them to be in, where they didn't overlap as much with the salmon, and that might be my salmon-centric point of view, because I maybe care a little bit more about salmon than I care about, I wouldn't care as much if they were eating other invasive species, like if they were eating juvenile largemouth bass or something like that, then I'd be happier than if they were eating the native juvenile salmon that were migrating through.
One more question? Well, mine is, what are the long-term trends for salmon? All this money and all, are populations increasing, decreasing? Are you making any progress with it?
It's not a happy prospect. I feel bad saying this on camera right now, but the amount of money that California is spending to save, and the outlook based on climate change for California waters, it's a losing battle, I would probably say. I mean, based on we're already at the southern end of their range, and I don't see Keswick Dam or more. I mean, we're taking out the Klamath Dam, so that's going to expose more habitat for coho and spring run up north, but that's better spawning habitat for them. I would say this Central Valley runs. I mean, fall run I think is in good shape, but the spring run and the winter run are, I think that's more of a losing battle.
Well, thank you very much.